The next thing we need to know is what will happen with the ammonia, because we know when we are degrading the amino acids, we are basically, previously we have just focused on the amino group, but we have seen during the process of degradation or catabolism, ammonia is also being produced. So what will happen with this ammonia, because we know ammonia is toxic to our body. So we need to know what will happen with this ammonia. So the ammonia that is formed in the body basically from amino acids are basically goes through the process of trans-deamination. Keep in mind, not transamination but trans-deamination. Where does it take place? In liver. Number 2, amino acid oxidases, the enzymes, and amino acids deaminases are also found in liver and kidney, so they can help to deaminize the ammonia. Deamination of physiological amines by the enzyme monoamine oxidase. Deamination of purine nucleotide, because purine, as you can see from this equation, purine is a nucleotide, which type of nucleotide which may, when they go through deamination or they go through degradation, may also produce ammonia. So in our body we have ammonia production from either amino acids or from amine or from pyrinucleotide or from pyrimidine nucleotide. Also, some bacteria plays roles in the production of ammonia, especially in the intestine, okay, and on the urea. So in our GI tract, basically these bacteria may cause ammonia production. Ammonia is also produced by glutaminase on glutamine. So these are basically our source of ammonia. Now, these ammonia mostly are excreted from our body, especially in mammals, because it is toxic to us. So usually what happens, they are converted into urea and then they are excreted from the body. Other vertebrae also do so. Some can excrete it as a form of uric acid, such birds and reptiles. Some fish can directly convert them into ammonium ion and then excrete, and so on. But we mammals mostly will excrete the ammonia from our body in the form of urea. Now this is how the disposal takes place. I've already told you that ammonia is toxic to our nervous system. So what happens basically? First thing that happens is that glutamate is formed. So the alpha ketoglutarate and ammonia are converted into glutamate by glutamate dehydrogenase, which then ultimately goes through the process of transamination to form a new amino acids and a keto acids. So first step is formation of glutamate. Second thing that happens is that glutamine formation takes place, specifically in the muscles and the brain. The glutamate that is formed now ultimately will be converted into glutamine by the enzyme glutamine synthase. In the brain, the glutamine is the major mechanism by which the ammonia is removed. In the liver, basically ammonia is removed by the formation of urea. The glutamine that we have in our blood circulating are ultimately all being deamidated by the enzyme glutaminase and are excreted. Third thing by which the ammonia can be disposed is urea formation. Okay, so let's again repeat. One way is that we can convert the ammonia into glutamate by the enzyme glutamate dehydrogenase. Then glutamate can be converted into glutamine. Okay, both glutamine are not toxic, but ammonia is toxic. Third way by which we can dispose ammonia is urea formation. The ammonia that is circulating in the blood ultimately can be transported to liver via two ways. Okay, first way is by the process of formation of glutamine. Glutamine synthase will take place or do this function. Second way is done by the muscles. This basically involves converting of the ammonia by the process of transamination into pyruvate and alanine. Okay, so the first mechanism, as you can see here, glutamate and glutamine. So the ammonia that we have in the body can be transported by in the form of glutamine, okay, which ultimately can be passed on to liver, Liver will do what? Liver will transfer or convert this glutamine into urea and ultimately help us to eliminate it from the body. In muscle cells, basically you'll see here the amino acids or the excess amino group or the ammonium that we have are converted into alanine. So alanine ultimately is taken up by the liver and converted back into pyruvate by the process of transamination. The pyruvate then can enter the carbon skeleton and help us to build glucose and so on. But the amino group that is obtained through this reaction of transamination ultimately will be converted into urea. Now in this process is known as Alanine cycle. Okay, so the one way is that in the liver we can pass on ammonia either in the form of glutamine or by the process or in the form of alanine. These are the two ways we can transport the ammonium into the liver. This is our glucose alanine cycle, as you can see here. So basically in our muscle cells, alanine and pyruvate are generated. By how? By converting the ammonia. The alanine is then passed on to liver. The pyruvate can be used in the carbon cycle. The alanine that is passed on to liver is ultimately converted also into pyruvate and urea. Urea then can go to kidney and then can be eliminated from the body. Pyruvate that is generated can be used to synthesize glucose. Okay, now if let's say we have kidney damages or let's say we have liver damages, in those circumstances our liver and the kidney cannot function properly. So usually they can handle the ammonia properly. Okay, so what will happen in those cases? In those cases, the ammonia level may rise very high, so it may be very toxic into our body. Okay, so it can lead to ammonia intoxication. Specifically, the ammonia that is produced by the bacteria in the gut usually is passed to the liver very rapidly via a portal system. We have hepatic portal system through which it is transported very fast because ammonia is toxic. We don't want to keep them for a long time in the circulating blood. So they are quickly converted, passed and converted to liver where it is converted to urea and eliminated. However, in certain circumstances, the liver fails to uptake this ammonia. For example, liver cirrhosis. In those cases, what happens? There are collateral vessels formed in the hepatic portal system. So the ammonia may bypass the hepatic portal system and may go to the circulating blood and may be toxic to our body. If we have ammonia intoxication, in that cases the glutamate that we have in our brain will be less or may decrease in number. Because why? Because glutamate will try to compensate. As ammonia level has increased, glutamate will try to protect our brain. So the number of glutamate in the brain cells will basically decrease. As a result of that, the GABA level also will decrease. And as these are being decreased, the excess glutamine that we have, because glutamate will be converted into glutamine ultimately, we know that, and so it ultimately results in several physical symptoms such as hyperexcitation, tremor, slurred speech, blurred vision, and ultimately may even cause death if we have ammonia intoxication. Okay, now comes another very important content, high yield, the urea cycle. We have seen that there are several ways through which we can remove the ammonia from the body, right? So one of them way was to convert them into glutamate and then glutamine. The second one was to form the glutamine in the muscles and the brain. Third one was urea formation. So urea formation, basically it refers to our urea cycles. Okay, so this is an important content, so you should put great emphasis and focus on this content. Okay, so let's begin. What is basically our urea cycle? The urea cycle is also known as you can see here, Krebs-Henseleit urea cycle. Ornithine cycle is also another name of the urea cycle. Ornithine cycle. We have just seen that urea is made up in the liver and then it is passed on to kidneys for excretion in the urine. Now how is it done and what happens during the whole reaction? Let's check out. So first thing, the two nitrogen atoms of urea are derived from two different sources. one from the ammonia, other directly from the amino group of aspartic acid. The carbon atom of the urea is supplied by the carbon dioxide. The end product, ultimate end product of any amino acid metabolism or protein metabolism is our urea. So you see here, urea accounts for 80 to 90% of the nitrogen containing substances that are eliminated from our body. There are 5 steps in urea synthesis and 5 enzymes are involved with these 5 steps. The first 2 enzymes are found in mitochondria. The rest 3 are found in the cytosol or cytoplasm. So this is how a typically urea cycle will look like, as you can see here. Ammonia, which are being acted by Carbamoyl phosphate enzyme, and ultimately this is our whole urea cycle. We are going into details about this, so wait for a minute. This is the structure of the urea. We have seen that its 2 nitrogen atoms are from 2 different sources. Okay, one is from the ammonia, another one is our carbon dioxide is produced by the carbon atom is provided by the carbon dioxide. Okay, and the second nitrogen is provided by the aspartic acid, basically. Okay, step 1. First step is to form carbamoyl phosphate. If you see here, carbamoyl phosphate, okay, the first step is to form carbamoyl phosphate. So who does it? The enzyme carbamoyl phosphate synthetase 1, in short we call it CPS1, found in mitochondria. So they basically will condense the ammonium ions with the help of carbon dioxide to form carbamoyl phosphate. And this process requires energy in the form of ATP. So specifically 2 ATPs are required for these steps to be taken place. The enzyme CPSI will require N-acetyl glutamate for its activity. Please keep in mind that there is another enzyme called carbamoyl phosphate synthetase 2. Okay, so don't get confused between CPSI1 and CPS2, because CPS2 is mainly involved in pyrimidine synthesis. They are found in cytosols. Their role is to accept the amino group from glutamine, and they does not require N-acetyl glutamate for its activity. So please know the difference between CPS1 and CPS2 enzymes. So this is what happens basically in first stage. First stage will form the carbamoyl phosphate. Okay, who is playing the role of enzyme here? Carbamoyl phosphate synthetase 1. So ammonia, the urea that we have, the ammonia with the help of carbon dioxide are condensed into carbamoyl phosphate. And for this reaction to take place, we need 2 ATPs. And also, to maintain this reaction, we need the help of N-acetyl glutamate. See the differences between the 2 enzymes, CPS1 and CPS2. CPS1 mainly found in mitochondria, CPS2 in cytosol. One uses ammonia, other uses glutamine. One is a part of urea cycle, the other one is part of pyrimidine synthesis. One requires N-acetyl glutamate, the another does not require. Second step includes formation of citrulline. This reaction also takes place in mitochondria, and the citrulline will be synthesized from the carbamoyl phosphate and ornithine by the enzyme called ornithine transcarbamylase. Ornithine is again regenerated and again is used in the urea cycle. Both the citrulline and the ornithine are types of basic amino acids, but they are never found in our proteins because we don't have, or they don't have the codons for it. Citrulline is transported to cytosol afterwards by a transporter system. So citrulline is also not found in any tissue proteins, nor in blood. However, citrulline can be found in milk. So this is the step 2, formation of citrulline. Carbamoyl phosphate and ornithine are ultimately converted into citrulline with the help of enzyme called ornithine trans carbamylase. Okay, the third step is formation of arginosuccinate. Arginosuccinate. Okay, now this will be done how? By condensing the citrulline with aspartate. The enzyme that will help in this reaction is called arginosuccinate synthase. And during this process, the second amino group of urea is incorporated. If you remember that there was two different sources of nitrogen. The first one was from the ammonia, and the second one was from the amino acids aspartate. So during this process, the second amino group is incorporated in the urea. Now this process also will require ATP, okay, because it involves breakdown of bonds. Okay. Step 4, formation of arginine or breakdown of arginosuccinate. Now this is carried out by the enzyme called arginosuccinase or arginosuccinate lyase. These basically enzymes, what they will do, they will break down the arginosuccinate to arginine and fumarate. Both of these are intermediates of TCA cycles. So during these steps, okay, this may create a connection between urea cycle and the Krebs cycle or TCA cycles, or directly to gluconeogenesis. So this is the fourth stage or fourth reaction. As you can see here, fumarate and malate, okay, they can be converted into oxaloacetate and aspartate. Remember that aspartate is regenerated in this reaction. Step 5 is formation of urea. The enzyme arginase is the fifth enzyme that is involved in during this reaction. They will break down arginine to get urea and ornithine. Ornithine that is generated from this reaction can again enter the urea cycle. Arginase will require carbon monoxide and manganese ions to get activated. Arginase is mainly found in the liver, while the rest of the 4 enzymes will be found also in other tissues. So that's why liver is considered as the main sites for urea cycle or urea synthesis. So as you can see here, liver can ultimately produce ureas. Because why? Because the source of this enzyme is only in the liver. Now let's check out some of the importance of this urea cycle. The significance. Energy cost. Only 1 ATP is required for this cycle. This cycle is related to also Krebs cycle. Okay, how? Why? Because of the carbon dioxide and also aspartate that is obtained from amination of oxalic acid and glutamate. Number 3, fumarate can enter the TCA cycle. C, sources of nitrogen in urea is either ammonia and the aspartate. Importance, forms arginine and forms urea. Liver have a higher activity of the enzyme arginase than any brain or kidney, because it is the place where the urea synthesis mainly takes place. Urea cycle also plays roles in the synthesis of non-protein amino acids, as we have just seen, ornithine and citrulline, which are not found in the blood or not found in any body tissue protein. Okay. Now, this urea cycle also is regulated by several means. Okay, how? By mainly regulating the enzymes. We have seen there are 5 enzymes involved in the urea cycle. I hope you remember that. So, the rate limiting steps, for example, the first enzyme, Carbamoyl phosphate synthetase. If we can regulate this enzyme, we can regulate urea cycle. Ornithine trans carbamylase or arginase. These 3 are all rate limiting enzymes. N-acetyl glutamate. We have seen in order for the carbamoyl phosphate synthetase 1 to get activated, we need N-acetyl glutamate. So via N-acetyl glutamate, we can regulate urea cycle. Activities of ornithine trans carbamylase. Okay, it will require ornithine. So if we can control the presence of ornithine, also we can regulate the urea cycle. The second way of regulating is to regulation through the flux through the cycle, flux of ammonia. That is the amount of ammonia that is produced to this cycle. So either from muscles of alanine or glutamine, or by metabolism via, okay, if we have a decreased metabolism, we will have a decreased urea cycle and so on. If we have a decreased amino acid degradation, we have decreased chances of urea cycle. It also can be regulated via availability of ornithine and availability of aspartic. And obviously the last one, changes in the level of enzymes. We have seen arginase, the most important enzymes, is required for urea formation. If we have someone who have a protein rich diet, okay, so in them the enzyme biosynthesis rate will increase compared to anyone who have a low protein diet. So if we can change the level of this enzyme, we can also change the regulation or change the control of the urea cycle as well. Now in urea cycle, sometimes defect can happen, and if so it happens, they can lead to hyperammonemia, excessive level of ammonia in the blood. And if somebody have no activity of urea cycle at all, total absence, it can lead to birth just after even, it can lead to death just after birth even, in people who have defects in urea cycle. So what kind of defects can happen? See here, number one, hereditary hyperammonemia. Patients are born with deficiencies of urea cycle enzymes. The enzymes, as you can see here, okay, can lead to certain defects, may lead to hyperammonemia. Ornithine Carbamoyl transferase, X-linked Carbamoyl phosphate synthetase deficiency may cause it. Citrullinemia, Arginosuccinate Citrulline, this kind of problem can happen. Okay, or N-acetylene glutamate synthase deficiency may also happen. Second one, acute hyperammonemia, not born with, after born you have developed it. So it can be due to liver diseases or maybe high protein diet. Okay, so you have excess amount of blood urea, but your liver is not working properly. So this can lead to high or excess level of ammonium in the blood. Now what happens? How do we treat these patients here? There is one principal protocol to treat for these patients who have defects of urea cycle. Okay, so what happens? We basically will give oral drugs that will help us to reduce the level of glycine. And if we are able to success in this, they may help us to deplete ammonia. How? By synthesis of glycine. If we have a low level of glycine, ammonia will be stimulated to produce glycine, so they will be used up. So drugs that we use are called benzoate, or also we can use drugs such as phenylbutyrate and phenylacetate. In case of extreme hyperammonemia, we can use hemodialysis, okay, where your blood will be filtered and sent back. And also very important, we advise these patients to have a low protein diet. Okay, those who have chronic hyperammonemia, we advise them to have a low protein diet. We also give them arginine supplementation.